The Effect of Caffeine on Brain Entropy. Brought to you by Caitlin Messenger, Mason Valley, Citlali Fernandez, and Jalen Ross. Background information. Entropy is a measure of molecular disorder. The more randomly matter is arranged, the more entropy it has. Entropy is a characteristic of brain function. High entropy corresponds with a high level of information processing. The scientific question was posed because the scientists in the article recently discovered that brain entropy differs between individuals and the controls involved. The hypothesis is that an increase in caffeine would cause an increase in the entropy of the brain. It was expected that caffeine would increase the brain entropy since it's commonly known to potentially increase focus and awareness.
In order to test the hypothesis, the scientists developed, an experiment was conducted. The experiment consisted of 60 healthy adults, 30 males and 30 females, between the ages of 19 and 31. An exclusion applied to the subjects that had an abnormal structural MRI, a history of head trauma, neuropsychological problems, and subjects that drank more than 1 cup of coffee in the past week. All subjects attended 2 imaging sessions, one with caffeine in their system and one without. On the first day, half of the participants took a 200 milligram caffeine pill and drank water. They waited 30 minutes after taking the pill to get scanned. The other half of the participants drank the same amount of water and waited for 30 minutes before taking their scan. 24 hours later, the participants returned and did their second scan. The scientists used the 24-hour period to allow the participants that took the pill the first day to fully digest it. The participants that took the pill the first day did not take it on the second day, while the participants that did not take the pill on the first day took it on the second day. In each session, a structural MRI, a RSF MRI scan, and an arterial spin labeling perfusion were taken.
In this scientific experiment, the control group was the people with no caffeine. The independent variable was the caffeine given to the people. The dependent variables was brain entropy, cerebral cortex, lateral prefrontal cortex, the DMN, the visual cortex, and motor network. The experimental variables was caffeine, brain entropy, cerebral cortex, lateral prefrontal cortex, the DMN, visual cortex, and motor network.
The results the researchers compiled from this experiment were that brain entropy increases after ingesting caffeine. This increase largely occurs in the prefrontal cortex, lateral striatum, visual cortex, and motor area. This increase of brain entropy indicates that in the resting brain there is an increase in information processing capacity. Therefore, the results do support the hypothesis. Also, there was a notable decrease in cerebral blood flow in the brain after the caffeine dose. The correlation between brain entropy and cerebral blood flow is the more increase in brain entropy, there is a lower cerebral blood flow in the brain.
Figure 1, particularly A, shows that caffeine has caused the brain's cerebral blood flow to decrease. This is signaled by the differences in the color blue. The figure also shows the decrease in blood flow in different axial slices of the brain. B in this figure is the 3D model of A. It shows the left, top, and right of the brain to show the decrease in cerebral blood flow in different colors of blue. Figure 2 shows the increase in brain entropy in the cerebral cortex due to caffeine. The caffeine had the highest increase in brain entropy in the lateral prefrontal cortex, the visual cortex, motor network, and the DMN, which is the temporal cortex, precuneus, and calcarine sulcus. Particularly A in this figure shows the 2D map of caffeine's effect on brain entropy. The red means higher brain entropy after ingestion of caffeine, while blue shows lower brain entropy after ingestion of caffeine. And particularly B shows the 3D representation of A, the left, middle, and right sides of the brain. Table 1 shows the increase in the percentage of brain entropy after caffeine was ingested. On the left side, it shows specific brain regions that are being monitored, and on the right, it shows those percentages of brain entropy. The percentage of brain entropy is being calculated by using the caffeine ingested condition minus control condition, all being divided by control condition. So in the DMN, the relative increase after caffeine ingestion was 16.09%. In the visual cortex, there was an increase of 14.48%. In the lateral prefrontal cortex, there was a 7.7% increase. And in the motor network, there was a 8.13% increase after caffeine was ingested. Figure 3 shows the very limited negative correlation between caffeine-induced cerebral blood flow and brain entropy. The areas that showed the negative correlation include the motor area, left calcarine sulcus, right superior temporal cortex, and the precuneus.
This figure shows the experiment design. It shows that 60 subjects were chosen, 30 males and 30 females, the age being 23 plus or minus 3 years. Then there were two groups, Group A and Group B. Group A on day 1 had caffeine and scan, and on day 2 just had a scan, while Group B on day 1 had a scan, and on day 2 had a caffeine and scan. Then they took the fMRI data, the functional magnetic resonance imaging data, and then did paired t-tests to compare the brain entropy and cerebral blood flow, and then also calculate the correlation between the two. In conclusion, the study shows that caffeine causes a remarkable CBF reduction as well as a BEN increase. These results suggest that caffeine does increase brain entropy. Some shortcomings of the experiment is that the scientists only had 2 groups to experiment with, one control and one experimental, rather than having multiple experimental groups. Something they could have done better is to have other groups with varying degrees of caffeine. Some strengths of the manuscript are that scientists explained everything that happened in the experiment thoroughly, including imaging parameters and data processing. They also included many figures which can help people understand the data better.